Let's just see. Haha, you all think it's going to be the red, most widely red. Yeah. Just here. So this is my 0th order.
That's our centre line, yeah.
So if I could ask you to draw a cross where the blue light is. Let's put the green on.
Green with some little triangles.
Okay, and now let's look at the red. All of you who looked on the widget at home who said the red would be the furthest away, give yourselves a pat on the back and take a little sip of your cup of tea.
Well, we can investigate the properties of visible light. So, so here I have a DVD. It's actually a level 3 electromagnetism DVD. You can use any DVD you like. And what I'm going to do is find my torch, and I'm going to investigate the properties of the visible light that is emitted by this torch. So if I shine this torch at the surface of the CD, what you see here is the light is being dispersed. So you can see all the beautiful colors of the rainbow. So what we have here is the white light being spread into all of its respective colors, and it's made up of different wavelengths of light. So you've got a slinky there that will help demonstrate this. So electromagnetic waves, so if we go here, so electromagnetic waves are transverse waves. So what this means is that the wave motion, so if I wave this, so the oscillations of the wave, so I'm sending energy through the wave from me to Rachel, but the oscillations of the wave are traveling in a perpendicular direction to the way the wave is traveling. So these are transverse waves, and electromagnetic waves have electric and magnetic fields that oscillate perpendicularly to the direction of motion. The case of longitudinal waves is where you have the oscillations in the wave traveling in the same direction. So here, this is how sound waves travel. So they travel in, so if you push that, so you see that the ripple is passing through the slinky, and so sound waves travel through a medium, so they travel through air, and the oscillations of the molecules in the air are in the direction of the propagation of the sound wave.
So this is a 16-inch diameter telescope, and there is a mirror at the end, as you may see from the reflection. It is so big because it needs to collect a lot of light. The sources in the sky, the stars, galaxies, nebulae, are very faint, and in order to improve on what the naked eye does, we need to have a much wider opening, much more light gathering power compared to what the pupil, the tiny pupil of the eye does. That's
one of the advantages of radio telescopes, of course, is that you don't need a clear sky. Here it's working on a cloudy day, it's quite windy, and of course it works during daylight. So I'm going to start off by taking you live now to the interface for Arrow. And we've had the Arrow telescope working away, tracking the sky, scanning away, and it's gradually moving around, tracking a part of the Milky Way that we've aimed it at previously. And I can take you now to Arrow. This is the control interface that you will be using. And what I'm going to do, you can see here on our plot of the sky, plot of the Milky Way, this is the crosshair showing where Arrow is aimed. We've got the coordinates down here. And what I'm going to do is to live set a scan running. So we are now making a measurement with Arrow live, recording emissions from hydrogen gas within the disk of the Milky Way. And fantastic, here we go. We've got a nice peak. In fact, we've got two peaks. So we've got signals possibly from hydrogen in two different parts of the Milky Way, perhaps at different distances. Pirate, on the other hand, is an optical telescope based out in Tenerife. And this is Pirate in action as night falls. You can see the telescope's busy, pointing to different objects. We've got the night sky moving around, so the telescope has to track those various objects. We're now live at the control interface to the Pirate telescope. So those of you who are doing Pirate will be using this control interface. You've got lots and lots of readouts and controls on the left-hand side here, and we've got the live webcam, Now in Tenerife it's not quite dark yet, they're a lot further south and a little bit further west than we are. So the telescope dome is about to open and it will start its evening schedule of observations. We can see here the Pirate telescope dome in the foreground. This is filmed from a webcam up at the top of a mast. We've got here an all-sky camera which is showing us where there are objects visible in the sky. We've got a bright object here, which is probably the moon here in the south, and the red dots are indicating where we're expecting to see stars. So as it gets dark, those will turn green, showing us where the stars are. The ray is going to be visible at any given time. So you will be controlling, in your observing sessions, one of those telescopes in real time to make those observations, to answer that question of what is the structure of the Milky Way, how does it work, how do we know that it's that spiral shape. You'll actually be helping to map that out. We now need to have a plan, because reasons for doing an experiment, we could point the telescope anywhere and get beautiful images of the cosmos, but we want to be a bit more organised if we're trying to answer this question, what is the structure of the Milky Way? And one of the first things that we could think of to do is to look at other galaxies. So here's an image taken by Pirate of the Andromeda galaxy, one of our nearest neighbours, M31 galaxy. And you can see here, of course, we don't know for sure at this moment whether or not our galaxy is going to be exactly the same as this, but if we see the same kind of materials and the same kind of structures, looking at other galaxies can give us a clue as to what our galaxy might be like. So we can see here we've got the central bulge, we've got a disk of luminous matter, stars and gas and dust, and we've got these wonderful dark dust lanes. So that's quite encouraging that we're seeing the same kind of features looking at another galaxy from outside, and there's even clusters and so on. You can see globular clusters and so on in and around. We can see areas of newly formed stars within that Andromeda galaxy. So it's actually quite reassuring to see that we've got the same kind of material and the same kind of objects in a companion galaxy, in another nearby galaxy. So we can start to make some comparisons with the structures that we see in our own galaxy. But of course, to answer the question in detail, we need to make measurements from within our own galaxy using the Pirate telescope to look at those clusters, the globular and the open clusters of stars. Looking out at the cosmos, light comes into there. Your raw data comes in the form of images, and here's an image of a globular cluster, and what you're going to be doing is analysing individual stars within that image, categorising them according to luminosity, colour, brightness, and plotting those parameters against each other to come up with a process data, which is this colour magnitude diagram for your cluster. And from that you can infer things such as the age of the cluster, the distance, those kind of things, again helping to map out where those clusters are within the galaxy. And here I've got a simple circuit with a capacitor and a resistor connected up to a battery, and I can charge the capacitor up simply by closing this contact here, and we can see I've got a voltage readout of what's happening. I've released the contact again, so the charge is draining away from the capacitor, the voltage is dropping, and I've got an LED here which is providing a visual indication of the voltage level on the capacitor. So what's happening here, you can see the digits are changing more slowly, is that the rate of change of the voltage is slowing down, and that's what we want to model. And in order to understand that, experiment again, but this time I'm going to start the timer when I release the contact. So we'll start the timer now, starting at 9.37, and you can see the voltage is dropping quite rapidly initially. The LED is still lit. As the charge drains away, the voltage is dropping, but the rate of change of voltage is slowing. This is what we mean by an exponential decay. So we've learned something important about our system, but in this particular system, maybe the capacitance is a little bit over that 20% because we just went a shade over that 38 seconds.
Today, who's busy carrying out a task, a pick and place task that you'd find quite typical for robots in industry today. One thing I should say right at the outset, that this is very different from many industrial robots, and that allows me to stand near it without being in danger of being maimed or killed or whatever. So one of the things that we'll be talking about during the session is analysing this robot in terms of its safety. This robot is called Baxter. It's made by a company called Rethink, who unfortunately have just gone bust recently. It was sold as a collaborative robot. So that's actually one of the things to have a look at, to think what we mean by a collaborative robot. So let's have a look at the results coming in on the widget and see what people are saying to how humanoid the robot is. And so nobody's saying it's fully humanoid, definitely, and generally they seem to be quite low. Well, okay, let's have a look and think about how that is overall. So one immediately humanoid thing is that this is an armed robot. It has two arms, just like I do. Geometry of a robot arm is very often rather similar to the geometry of a human arm. So typically they would have something that corresponds to a shoulder, an elbow and a wrist. And so roughly what we have down here is something that corresponds to the shoulder joint. Here is roughly the elbow, and here is something like the wrist. Another way of thinking about the number of motors in there is to think about, in terms of control, what things do you have to control, which in this case would be 7 different motors, 6 at a minimum, 7 in actuality. And one way of talking about that would be in terms of degrees of freedom. So we can talk about this as being a 6 degree or a 7 degree of freedom arm. Now the program that I'm running here is going to let me control each motor individually just using the keyboard. So I'm effectively doing a bit of remote control on it. So by pressing one of the keys, I can drive one of the motors. It's the rotation down here at the bottom, and going the other direction, the same one coming back, I'm controlling that motor. In the wrist as well, there is a hidden accelerometer which will which will sense the angles of pitch and roll and so on. So the same sort of technology that you would have in a mobile phone that will detect the angle you're holding at is actually hidden inside the wrist here as well. At this point, I think it would be appropriate to talk about Asimov laws. So we have another widget, which is, do you think this robot obeys Asimov's laws? Quite well up towards that end. So people are thinking it does obey Asimov's laws. And actually, there are some wrinkles in here which mean that it's not quite right. For a start, asking it to always preserve human life means that it should have some concept of what a human is. And actually that's not the case, is it? It doesn't know that I'm a human, so it's not distinguishing me from any other object in its environment. So what I'm going to be doing today is to do a little bit of networking with some Raspberry Pi computers I've got here. First of all, do some wired networking. So I'm going to connect my laptop to Raspberry Pi using some wired cables. But then I'm going to go on and try and do a little bit with some wireless networking. Typical Raspberry Pi, which I have got connected up with a keyboard connector and a mouse, and over here the monitor. And so I'm using it as a standalone computer. So point out now immediately though are these 2 more Pis that I've got over here, which have got some things attached to them. So these are Pi Zeros. And this one has, let me turn that around a bit easier to see, This has some environmental sensors attached to it on a separate board that plugs in on the top. This one has got a series of LEDs which can turn on and off, make a little bar chart. But before I do anything else, I really need to make a bit of a plan. So if you're doing any sort of networking, you need to have some sort of plan of what you're going to do. So I have my laptop connected to the hub, and I have the Pis connected to the hub. The other thing I need to know is some names for all these computers. So each computer will have to have its own name. So my convention will be that this one here is John's Pi 3 yellow. So that's my name for that one. Now I also need to think about the IP addresses. So IP addresses I'm going to set manually for this. I'm using IPv4, so these will be dotted quads, so 4 numbers, dots in between, to 168. And then there are 2 more places in the dotted quad that I can fill in. I'm going to pick something slightly random for the first part, 42, and then leave the last part to vary for the devices in here. So I'm going to set that 0 for the network. Online, so I'll open the command line and use the command ifconfig to see. And there we have. So if we got that on screen, you can see the address that I did set, 192.168.42.250. And to ping my laptop from here, 192.168.42, and the address of my laptop was .240, and hopefully I can ping in the reverse direction. There we are, that confirms it.
And as soon as we start to mix them, just like the rungs of on a then, whoops, there we go, we end up with a gel.
Borax in solution forms an ion that makes weak bonds with the OH groups in the polymer chains.
And you can see as Rob pushes it down, you can see like a puff up the balloon.
So we're warming up to room temperature. There's a couple
down. You can see we put in some earlier on.
So we're warming up to room temperature. There's a couple down.
So you can see how the balloon is self-inflating.
The experiment is this. We're going to suck the air out the flask, form a partial vacuum. In other words, reduce the pressure that is around the gas in the balloons. And the question we're going to ask you in the widget, which should appear at any second, there we go. When we pump the gas out of the flask, would you expect the size of the balloons to increase, decrease, or stay the same? Okay.
92% think that the balloons will increase in size. Oh yes.
I think there's no doubt.
There's no doubt about it.
So we agree with Robert Boyle, I think.
That's right. And if I release the pressure, you'll notice what the problem is. The balloons shrink back to their original size, so they won't stay at that size.
Thank you, Mike.
Now we're going to look at close packing of atoms in metals, and you can see that we've got a tray here where I've put in a row of atoms to begin with, and I've just started putting in the second row of atoms. And once we've got this second row of atoms in, we then have 2 possible options for where we can put that third row of atoms.
Plus, we're actually going to add some ethylene diamine. First of all, we're going to do it in a 1:1 molar ratio, the complex represented by this model. So what we've done is replace 2 waters with 1 ethylene diamine. Let's go a step further. And we'll now replace 4 of the water molecules with 2 ethylene diamine ligands. Hopefully, we'll form this complex here. Okay. So this time we get slightly deeper blue?
Yep.
Okay. So finally, we're going to replace all the water molecules, in this case with 3 ethylene diamine ligands. Remember, it's always an octahedral complex. We're not changing the geometry. We're just simply substituting 1 ligand for another one. Clearly is a much more purpley violet colour. So there we go, one example of the colourful chemistry of the transition metals. But it's quite simple. We're actually going to use this molecule here, which is glycine, or the glycinate ligand. It's an anion formed from the glycine, which is effectively the simplest amino acid, and that will actually form a bond to copper. In actual fact, it goes a step further than that. It'll form 2 bonds simultaneously to the copper 2+ ion.
Just waiting for another moment, so you can check the temperature on that one.
I'm nearly there.
Oh, this is going to be, I can say whether maybe to keep it on, to keep it at 70. Maybe just turn it down
It's
a bit.
going to, yeah, it's going to go up. Isn't that well up?
Allowed to cool at room temperature, and so the precipitate will form.
Okay. So we've got that attached to the pump first. Excellent.
We haven't got the solid yet, have we?
Rob, just in anticipation for doing suction filtration, should we have this connected to a stand to help secure
actually,
it?
that's a good idea. Yeah, because it's quite a top-heavy bit of apparatus.
I mean, these are very close.
To do the very end bit, would you be able to use anything like a little bit of ethanol? Would it just to rinse the
Yeah, if you just tickle it around with a bit of ethanol, swirl it into a kind of slurry, and then just whop it in. That's great. Plenty there. So that is.
Try that one.
So for those of you watching, this is isomer A.
This is isomer A before it's dried.
Before it's dried, yeah.
But it's not too different in colour when it comes out.
No, it's pretty much the same, yeah. Yeah, so that's our filtrate, which we're going to use in the next part of the experiment. You've got your isomer A. So we started out with this equilibrium mixture of A and B, okay? Now, if you think about it, if you're actually taking A out of the system at an equilibrium, what's it going to do? What's the equilibrium going to do?
It's going to try and shift. It's going to try and shift. Well,
Yeah,
it's actually trying to-
yeah. I'm sort of thinking along the lines of Le Chatelier's principle. Does that sound familiar?
Yes.
So,
It's very sensitive, isn't it? So
0.071 grams.
well, actually, it would be an interesting experiment because isomer A does
theory, it
naturally
should still
convert
do
to
it,
isomer.
yeah.
Oh, yeah, absolutely.
So why do we add the glycine?
Swizz it round. You're all stuck at the bottom. Swizz it round before you come up.
So what we have here, so some of you might have seen this setup before, for example, if you came to the Level 2 lab school. We've got essentially a setup involving a burette that's contained sodium hydroxide in this case, and some ethanoic acid in the beaker at the bottom there. Okay. Actually, this is the exact same setup that we're going to demonstrate to you of the remote apparatus. Okay. So the pH meter is measuring the pH at each moment. So I'll add a bit more on sodium hydroxide, and as you might expect, the pH rises a little bit. Okay. So I add a bit of sodium hydroxide. It raises the pH a little bit. And this is all to do because I'm changing the amount of A- and the amount of HA present. What we end up doing with this data is basically constructing a titration curve.
As much as we might like to, we can't send you a burette and all of this apparatus through the post. So the next best thing that we've really got for you to do is to do a remote experiment. So this is an auto titrator that we've set up in one of the other labs in Milton Keynes.
We have our reservoir here of our sample, a second reservoir of our titrant, and these are transferred by these tubes into the dispensing burettes. They're then transferred into the titration vessel, but you'll be controlling it through the Open Science remote access interface.
So Nick, have you got our completed titration curve
We
now?
do. You can see that we have got a good curve here, and you can see that we have a strong inflection at around 0.98 mils and another inflection occurring at about 1.04 mils. So somewhere in between here we have our equivalence point, which I would suggest probably around here around a pH of 8 or so. So that's about 1 mil volume. So given that, we're looking at the half that volume to get our 50%, a half pint equivalent, and that's around a pH of 4.7, maybe a little less. It occurs here.
I'm going to do a sort of a bit of demonstration of photosynthesis, which I've got to take just check that everything is going okay here on that. A setup here, this is kind of pretty similar to the sort of thing that I use in my research when I'm studying photosynthesis in the lab or in the field, and basically it's a chamber that has a plant sample inside, which in this case is some moss that I've collected from outside, and this is connected to a infrared gas analyzer which measures the CO2 concentration in real time, and so air is circulated through this chamber and we can see how the CO2 concentration in the chamber is changing, and this is connected to a computer which we'll look at in a minute. So hopefully now we can go and have a look at what's happening in the plant chamber here, and I've got up here a real-time figure of showing the CO2 concentration in the chamber, and what you can see there is that although it might not seem very strong, there is a sort of a great, well there was a bit earlier on and now it might be just settling out a little bit, but there is a gradual trend down in time of the CO2 concentration, and this is a result of the moss that's photosynthesizing inside the box here. Without the light on, without the moss inside there, the concentrations were up over 800 ppm or something like that we were recording earlier on. And now I'm going to try and see if I can turn out the light and see if we get a response that is obviously going to involve the reverse of photosynthesis. So now we'll plunge the plant into darkness. Look, we're starting to see the response there. That's
looking good. But what was really noticeable there was as soon as you switched the light off, it was such an immediate response.
Exactly. Yeah, exactly. Yeah. So we were we'd kind of run down to what we call a steady state situation in the chamber in terms of the CO2 concentration, and that gives you an idea of the amount of CO2 that the photosynthesis was pulling out of the atmosphere there.
Now, Rachel, you've got the worm experiment. Well, we're going to set that up first because it takes a little bit longer.
Yeah,
so tell us a bit about it.
Okay, so as I mentioned before, we're interested in how animals change their behaviour in response to infection.
Yeah.
And so the experiment we're going to do tonight is going to be looking at movement. So if we look on the microscope over here, I've got some worms set up for us to take a look at.
Oh, that's great. Look at that.
So these are just normal worms. We call them wild type. So they're the worms that we would find in the wild. And you should hopefully be able to see if they are, but they're not moving very much. They've been sat on this microscope for a little while. Okay, you should be able to see the one in the middle starting to move.
Oh, look, there's one
now.
from the one on the
and
left.
they're moving with kind of a sinusoidal wave-like or S-shape-like movement.
Right.
So if we compare that to animals which have got a mutation in a gene which is important for movement.
That's not really S-shaped.
It's
Rachel,
not really. It's very, very curled up. And you can see on the plate underneath it, you can see lots of little tracks that are very, very curly.
Yeah.
So this animal's got a mutation in the gene that's important for movement. The experiment we're going to set up today is an experiment that we use to kind of give us some of those numbers.
Yeah.
It's called a dispersal assay. We use one of these slightly bigger plates.
And that has exactly the same stuff on it as the other one,
exactly
yeah?
the same stuff in it. And what we do is we put a ring of the worm's food around the edge. And then in these tubes here, I've got a couple of different worm strains. So one of them is our normal wild-type worm strain, the one that had the normal sort of S-shaped movement. And then this one here is a mutant, which has a mutation in the gene that's required for movement.
Yeah.
What I'm going to do in a second is I'm going to put a few of the worms into the centre of these big round plates.
Yeah.
And then we're just going to leave them for 20 minutes. And at the end of the lab class, we'll come back and see what's happened.
And see where they've moved. I wonder how your worms are doing, Rachel.
Should we take a look?
Should we take a look?
So if we go back to the microscope and have a look. So I've now got my dispersal assay plate under the microscope. And it's a bit difficult for you to see because the actual field that you can see under the microscope doesn't cover the whole of this plate. But if I go to the middle, you should see these are my wild-type worms, and we can still see quite a few of the worms in the middle of the plate. But if we move outwards, we start to see a few less. And when we get right out to the edge of the plate, we don't really see very many. You'd probably just about be able to see when we have the shot of the microscope itself that there's some tracks there, and we can see some tracks of them moving out to the edge. And if I take a look at my mutant worms now, let's have a look and see what happened with those. So I think you should already just about be able to see that there are less tracks coming right out to the edge of the plate.
And that little bit in the middle that you can see, is that the worms?
That's
Rachel?
the worms,
Yeah.
those little spots and dots in the middle. And if we move out to the edge, again, we can see less. So we can hardly see anything there. So because that's a little bit difficult to see, I've got some time-lapse movies that I've taken of these animals. In an experiment that I did earlier. So what we're looking at here
They're quite fast.
They are. It's a time-lapse experiment, so it's sped up a bit. So they don't normally move this fast. So this is our wild type, our normal animals. And the dot in the middle is where I put the worms at the start. And you can see as the movie moves along, you can see them spreading out from that dot in the middle.
You can.
And moving out towards the edge. So those worms are kind of dispersing quite well. But if we take a look at our mutants, you can see...
Oh my gosh, they look very different,
don't they? So
Yeah.
they don't really seem to be moving as far away.
They don't seem to be moving very effectively
No,
either.
they don't. They seem to be going round and round in circles a little bit, don't
Yeah.
they?
So the mutants, where would you have put them on the three choices that they had, that everyone had?
Well, personally, I would have predicted that they would stay in the middle. Should we take a look and see
what
what
everyone else has done? Yeah.
So
more at the edge, actually.
Most people have gone for more at the edge. Although it seems to be fairly evenly split between more in the middle and more at the edge. And everybody thought that there would be a difference, or almost everybody. So that's really good.
Yeah. So you've got a digital version of a real microscope. Now we're going to show you the real microscope tonight and some of the real slides that have been digitized for you. And you're going to use this digital microscope to look at some different tissue types and some different cells and to do some really good quantitative research work on it. There'll be quite a big focus tonight on microscopy, and we'll come to the light microscope a little bit later. But first of all, I think before we start to look at the really small structures in the cells, it's really important that we look at the bigger tissue that the cells come from.
You forget that what you're really also looking at is a whole piece of tissue,
Yeah, absolutely.
a functional unit that sits within the animal. What we're going to look at is actually give you a view of the whole tissue, and to do that we're going to use a scanning electron microscope. This is sat here on campus and I can control it remotely from here in the lab. Before I do that, I'm just going to go to show you what the sample is held on. The samples are probably about 0.5 millimeter long. This is a small little stub, what we call it. It's a piece of metal. Sat on that metal in the actual electron microscope is a lump of tissue that's probably about 5 millimeters in size. That whole stub is about 1 centimeter across, just less than 1 centimeter. And this is a live image. Just to convince you it's live, I'm just going to slightly move slightly and just let it adjust. So this is a picture of a piece of adipose tissue that actually supports and surrounds a lymph node, but it illustrates quite nicely what we're looking at in terms of cells and the outside structures of tissue. So we looked at the microscope and we came out with cells that were somewhere between 50 and 70 microns across on a very crude measure.
Yeah.
How is sectioning going to influence how we see those?
Right, so first of all, the first point is the size of the cells. So you're talking about something that's significantly larger than our sections. Our sections are slices that are about 8 microns thick, so they're very, very thin. When we call them thin sections, they're very thin. So that essentially means you're going through the tissue or the cells in cross-section. You're going to actually cut through them when you section at all sorts of different angles, which will affect what you see. But I think you've got a much better way of showing the students.
Well, I have. So the really keen-eyed amongst the viewers would have spotted an array of foodstuffs. So let's assume that this is about 50 microns across, so long. And if you think of lots and lots of piles of bananas all sat on top of each other, what we're talking about doing is making a section that's probably a very small section across a single cell. Now, they're all in different shapes and orders, and if you imagine a section, I'm going to use a knife here. If you imagine that this cell was actually just cut right at its end, and I'm going to do this very carefully and not take my finger off live. What you would see in cross-section would be something that's relatively small, like a small circle. It could be, though, that the knife sections obliquely across it. I'm going to do this on the bench because it's a little bit more dangerous. So what you end up with are sections that look a lot wider and a lot more of an odd shape. So in fact, the same cell might appear as a very small cross-section, if
Yeah.
it's just happened to be. It may appear as a cross-section. And the same goes for the things in a cell, like
Yeah.
the nucleus, for
Well,
example.
I mean, if you look at the middle of that banana, Mark, one of them looks like a dot and the other one looks like quite a long extended line, and that's no different to cell nuclei, really, if you cut through them at strange angles.
So we have a
Best
close-up of a
focus.
microscope here, and what we're actually seeing is light from the bottom going through the
Yeah.
slide, and then the objective, in that case, is a times 40 objective.
Yeah.
You have a similar set of objectives on the digital microscope
Absolutely.
as well.
And this is one of the key things you have to take into account. You need to actually be aware of which objective you're using and make sure you don't change them between your counts, because that will obviously affect how big and how many cells are in your picture. Part of this topic is learning how to count like a biologist. Bring a grid down. If I just click that over.
By the power of magic.
By the power of magic and high technology, we're going to be focusing on the nuclei. So if you can see on screen, I'll just point, that, for example, is one of the nuclei. It's the darker. Remember I said the stain is likely to be more attracted to the nucleus of a cell. We're going to be using a specific protocol to count nuclei. So, I mean, we're going to count within the grid, and what we're actually going to do, we're absolutely going to count everything that does not touch the edge of the grid, because there's no question about that. That resides within the grid. But there are some things where there's a questionable area, and I mean, you get little bits. There's a tiny, tiny little bit of a nucleus in here, and again, there's a nucleus up here that sits part in and part out of the grid.
So we have a grid that we always use.
Yep.
We always use the same magnification.
Yep. Yep.
So what do we do when a nucleus sits on the boundary of the grid?
We have a rule.
We have
Mark.
a rule,
We
okay.
have a rule that we apply, and it's really important that you apply this rule in the same way every time you count, so that you're systematic and consistent. And what we do for our rule on 288 is that we count anything that is touching the right-hand side or the top margin.
Okay, so something like this is just in, but
And
almost
touching.
close and touching. So
So
this one would be
We
in.
would count, but we
Okay.
don't count anything that is either on the left-hand side or the bottom margin.
And I'm going to show you and demonstrate a live event which is regulated by calcium here in this labcast.
So how are we going to see autophagy tonight?
So tonight we're actually going to use human cells. So I've got an immortalized human cell line on the microscope here, and a PhD student of mine is working on autophagy, and she has prepared these cells for us. And the advantage of these cells is that they are bigger than yeast cells, so it is much easier to visualize processes which are going on inside these cells. So that is an image. We are exposing the cells to white light, and here you can see the outline of the cells. Oops, if I get the mouse to work. So here you've got the outline of one of these cells, and in the middle of the cell you can see slightly oval-shaped the nucleus of the cells. So the cells look a bit like a fried egg, with the yolk being the nucleus.
So these cells we're looking at, I can sort of make them out. They're effectively transparent. They're live cells. So how are we going to see the organelles inside them tonight?
So if we want to look at the organelles, we have to use a little trick. We won't see them in white light, but these cells are engineered to express the green fluorescent protein can be seen under the in this microscope if we excite the cells with blue light rather than with white light. I now increase the light and turn the blue light on, and I have to cover the cells to get rid of background light. And what you can see on the screen now is that we can see the same cells, but we can see the green fluorescence in the cells. And what you can see are these white green dots here.
So those dots are the actual live autophagosomes in the cell, just routinely cleaning up the cells.
Yeah, so the recycling processes happen in these vesicles, in these bright green dots. But another option of upregulating autophagy is a chemical induction. So we can actually add a chemical called rapamycin, which you might have come across in your studies. And I'm going to do that now. And we hope that at the end of the labcast, we actually see a larger number of these bright green dots inside the cells. So I have to take that little lid off. And I've got the rapamycin in here. So I'm going to take some rapamycin and carefully add it to the cells on the microscope.
Okay, so they're going now. So I will
They're
start
going.
the timer so we know how long we've been going. So, and we just leave, leave them sat there to
cover
incubate?
them. We just leave them and hopefully see more autophagosomes later on today.
Okay, great. So we've got the timer running. To check back in with our live cells, Katja, do we have more autophagosomes to look at?
We do have a lot more autophagosomes in here. So I think I did a little count earlier on when we first looked at the cells, and I had about 50 15 autophagosomes or so per cell.
Ooh-wee.
And now, let me
we
just
can cut to
focus
the live
on
image.
that, on the live image.
That's significantly more.
That is significantly more, I would say, even without doing a statistical test. And the big question is, can we actually see something moving in here? Because we promised you these vesicles are actually moving, but if you first look at the numbers, I think we've definitely induced autophagy and see a lot more vesicles per cell.
So anybody with sharp eyes might be able to spot them moving. I can't look at all of them at once, it's quite complicated. So we've just spent some time talking about how we are very systematic in terms of counting on a light microscope using a grid. So how on earth do you quantitate these types of events in terms of what we see? How do we count them?
So what I do here is, rather than counting a certain area, which you do with counting a grid, which is a certain area, is counting all the dots inside a single cell. So I define where a cell is, and here you've got the outline of one single cell.
Yes.
So I would look where is one cell, and then I count all these individual little dots inside the cell. Yep.
These are obviously cells in 3 dimensions, and the question, we saw very thin sections which are very clean. How do you record data in cells where things are moving around, but also it's in 3 dimensions?
What we actually do is, if we want to record the moving, we record movies. So we just take a live stream, record that, and see how the autophagosomes move through the cell. But if we want to quantify, we actually take still images. So we take images before we add the rapamycin or induce autophagy by any other means, count cells in the before treatment, and then 1 hour later or 4 hours later, we take more images of the cells and we count several cells so that we've got the average number of autophagosomes at different time points.
Okay, so it would be very nice to see some moving autophagosomes. I think we have a short clip of some that was actually collected beforehand, if we can see.
But you could see them moving, and we're going to load up a few more movies on the forum website.